decontamination for this core. And those of you who are watching, we're doing the core sediment sampling this way too. We're pushing the core samples in at the same time, because when you see when we pull the first one out, it will disperse some sediments around, and we don't want to contaminate this second sample with the
Fine with
sediments
here,
further down.
Yeah, that looks great.
Every time I see the manipulator come up with a core sample and you can see the cloud of dust coming out of the bottom, all I can think is quick, quick.
same here.
Yeah, because it could just fall out. I'm sure that's happened.
Once or twice.
This looks like some fairly cooperative mud.
Yeah, it's pretty hard. They go in really easy, and so you have to worry.
Nice.
Okay, close that box.
And those were sample numbers 82 and 83.
Roger.
You want to look at the suction sampler again? Pan it up. Come out of south, yeah.
Going back to dive, yep.
And then change the camera over to the jars. And we'll
He's asking if the suction sampler is the best way to get cup corals.
with these jaws, I think so, yeah. They're little tiny guys.
Yeah, they're pretty small.
Yeah, I think they'll go in there. I don't think you can grab them with the snaggletooth jaws. I can try, but
yeah.
if they were bigger, we could kind of cradle them. But I don't think they're big enough to, they'll just fall through the fingers, won't they?
Yeah.
True.
Okay. Switch to jar 1 and move the camera over and zoom in there. Might have to move closer. To the left a little.
Which one are we going for? Left?
I don't know. I'll have to zoom in. And
Which one do you want, Meredith?
I'm taking a look. Let me get that back to full screen. There was one right above the tube worm. That's kind of to the right. And then, yeah, there's this one that's right on the kind of lip edge of this rock. There's several. I'm looking for the scientists ashore to see if they have a preference on any of these specifically.
I need to zoom in. I can't see. That's better. And you can back off on your verts a little now. So causing the camera to shake. Like about 30%. Is that one just in the very right-hand side of the picture there?
Yep, that's one on the very right-hand side of the picture, right above the fluffy tube worm.
Yeah, okay, zoom out just a
Whoa.
bit. Okay, I can zoom in a bit. That's good. Okay, I'm like 50% there. Stretched out, so it might be a little sloppy. I don't need that bubble camera.
I was just looking at the hose, make sure it's
Yeah,
stretched
we
too far.
already been here, right? Oh, good. 50%.
Goodbye. Doesn't want to be slurped.
Polycute. Yeah, Polycute just said, I don't want to be slurped.
Yeah. So are we going to be slurping this cup coral, which is living in seems like a calcareous skeleton? We're going to slurp it out. Oop,
Oh, I'm just gonna
got that.
knock
We're
it
going to
off the
scrape
wall.
the whole skeleton off.
Okay, the whole thing.
Yeah. So there's a group at the University of Washington that is using these for some paleoclimate studies and modern climate studies. So they're sampling the skeleton and then looking at the isotope signatures in them.
Tooth extraction.
We need to move closer.
Roger.
Zoom out, Steve. Bring your head to the left. 45 degrees or so, maybe 90 degrees, and get up close. Can't be accurate there at the full arm reach.
Mm-hmm.
It's really sloppy out there. Trying to write neatly on the whiteboard while you're standing at arm's length from it.
With your pen on the end of a stick.
Yeah. That's good, Jake.
Yep.
Maybe
This
come
is
back
a
to your right
delicate
a little,
operation.
somewhere in there. Move to your left a bit now. Left more.
Left more.
We're on the camera on the left side of the what we're trying to get. So
Pushing.
you basically put the temp probe that's sticking out there.
Yep.
Put that right where we're sampling. Maybe just a little more to your left. Bring your head to your right a little. Yeah, now
Back's hitting.
hold that head and lateral left. Yep, somewhere in there. Probably parked the front left of the vehicle onto the rock there.
Right here.
Yeah, that'll work. Planter there.
Got her.
Okay, let's find another one. Should be able to. I don't know if I got all of the first one. You want to zoom in on that spot? Definitely wasn't in one piece.
Looks like there's a little of the base of it left.
Yeah. Turn that jam up a little bit. Suction jam. Pro 75. Go for the blind. Blind's great. That got, no, can't quite get in there.
Yeah, it's the handle is hitting the wall before the tube is to scrape it off.
What?
The handle end, like of the wire,
Oh,
that's what's hitting the wall, not the tube. I don't know how.
There seems to be one right below it to the lower right, right.
yeah, I'll go for, I can, oops, I can try a different angle here. Do you want the whole base there, do you?
If we can get it, yeah.
Yeah, not having much luck. It's kind of in a little nook there. And zoom out for a second, Steve. Just a little too close
There's
there. Bring your head to the right a little. Ladder left to bring your head to the right. And then back up some.
Yep.
Just a little more room. I actually have a control over here for that. Nope. You're on top of the rock now.
Yeah. So back a little bit more.
Yeah. There'll be some way you can get down in there. You were good down in there before, it was just a little too close. We can put the front of the vehicle parallel with that little wall in there and snuggle in there. Too close, mate. Yeah, right there somewhere maybe. Yeah, perfect. She gonna sit down there? It's not coming off.
Our our scientists assure us that if we can't get the rest of this one, that's probably okay, and we can try a couple of the others that look like they're a little more accessible. Like there's one in the upper left-hand corner that's kind of right on the lip, or further over to the right-hand side.
Yeah, I see the one way up to the left.
Yeah.
Up here.
Yep. And Lizzie's also saying that just to the right of the lasers, there's a dead one.
Well,
I'm not.
sure I'm looking at the right thing. Is this guy here?
Yeah.
I think the one below it is also a cup coral, is it as well?
Yeah, there's one just below the slurp, although it's definitely alive.
Yeah,
Zoom in there, Steve.
the one that's got its, what about the one at the bottom? Is that a cup
That's
coral?
a cup coral there in the center of the screen, isn't it? I can't tell.
Yes.
I'll go for him.
Do we, is the distinguishing characteristic of a cup coral that they have that knob, besides being shaped like a cup and being in solitary, that they have those knobby ends at the tentacles?
I don't know if that's distinguishable, like that's characteristic of the cup corals themselves. I think there are a lot of coral species that when they have their polyps out will have that.
There, on that rock.
My molars are hurting after watching.
Yeah.
There is one right below that looks like it's more
Yep,
likely
that
to
one would
bust
be
off.
fine.
Unauthorized camera movement.
What?
Teasing you.
Unauthorized camera.
I think you can reach over and hit him.
Can
Got that one.
kick his chair.
Oh, I
Are
don't
you
even hear
talking to
hear the
Jake?
comment, so I'm just gonna
Ignore me
pretend I
like
didn't
That
he
was
hear
a
it.
usually
good
does.
capture, that last one.
I don't know if that one went in the jar or not. Haven't been watching.
Yeah,
See,
I think it did.
was there one more there you were eyeballing?
If you go all the way to the right edge of the rock, right above where it joins the sand, there's one right out on the lip again.
Pen, right? Oh, I see him way down there.
Yeah.
Yeah.
There's also one right above the temp probe.
Yeah. Can I zoom in there, Steve? I like the one on the left. Looks like a easier spot to.
Go for both.
It's long jaws there. Not my favorite. think if they don't come with the first nudge, or
Oh, it
Try to
dropped
fall
down.
down.
zoom out a little, Steve.
All right,
Oh, it's, I can see it right there on the bottom.
that's
Yeah.
an intact one.
Or do you want the last one as well?
I'm looking online. If we can get it, let's go ahead and get the last one.
That was great. That's like tooth extraction and dropping on the floor and then being able to pick it up again.
Hey, you want to center the camera on him and zoom in a little while I make an adjustment here.
I don't know if I've seen that one go in yet.
No. Zoom out for a sec, Steve, please. Thanks.
What's our, what are we on, 50%?
I'm gonna go full jam for a second.
Yeah. Come on. Turn it off for 1 second.
Oh,
There it
that's
is.
that's a sponge that we picked up in the beginning. I think a little piece of sponge.
So. Okay. Full jam again.
Is that it?
This is
No, I don't
It's a
think
hermit
so.
crab.
No, not that
Is
one.
that
It was something
crab
else.
having the ride of his life.
Yeah,
I think.
there's some more stuff.
No, that's not it.
Is that it in the bottom right, that white spot?
I don't think so.
There it is.
Yeah.
Oh, that's
Yep.
it. Yeah, that's it.
Okay. Zoomy zoom.
So I'm trying to verify with the scientists through science chat. So we're collecting caryophilia right now.
Yep, they're pretty sure this is caryophilia.
Okay, great. Which is one of the cup coral species.
Yes.
And how many are there
Zoom
of
out a little, Steve.
that we might encounter? Well, we'll wait for them to finish this.
There's a fair number of West Coast cup coral species, but some of them only occur deep.
Oh, so Lizzie's writing down there's caryophilia, desmophilium, and balanophilia. Balanophilia are the orange ones, I believe, but I don't
Yes.
recognize the other names. And we should keep track of which species we're collecting for the permit.
That one's not coming.
Get the sawzall.
What's that?
The underwater Dremel.
Yeah, sometimes they're just on there like.
Yeah, that one's like the base was way wider than the cup coral, whereas the one that popped off, it seemed like the base was not that wide.
Daniel is saying that the Cairns paper on West Coast coral species has about 50 species of cup coral in it.
Oh
Yeah,
my
I'm
goodness.
gonna break the manipulator, trying to put I have full jam on it there. See how far it flew after it let go. Yeah. That was about it, was it?
Pan left.
One up and to the left, is there? You have to zoom in a bit. Okay.
So I see a couple more tucked right there on the left of the screen.
The orange guy?
Yep.
There's maybe one hiding right to the right of it, down in the...
Yeah. Next to that brittle star.
Yeah. I think that's another one hiding next to the brittle star.
Okay, zoom out for a sec, please, Steve. Thank you.
Lizzie says, maybe try just that top one that's not tucked down in the crack and we'll call it good for this site.
Okay. They're so little, I think they're pretty darn tough on there. Maybe it would be better to put the suction sampler down and try and pop it off there with a finger and then pick it up.
You think that would work?
Oh, this is not working. Zoom in, Steve. I don't know if I broke
Lizzie
it.
says she thinks she got some pieces.
Got a piece of it on top of the white spongy.
Yeah, I think that's a piece of it on top of the white sponge
Yeah.
there.
Uh,
Yep. And Alex says, really happy. Thank you, everyone.
okay, I'll do one more scrape here on the way out.
Just behind the manipulator arm, that little yellow twig is actually a newly described species of Plexarid gorgonian called Swiftia cordalbankensis. And this might actually be I think we found it in the Channel Islands before, but it's known from further up. It was described from further up the
You want
coast.
to zoom in on what she's talking about there?
You just, zoom straight in. That yellow twig right at the top of the screen, just between the lasers, is Swiftia cordalbankensis. It was described last year.
From Cordell Bank.
Yep.
Oh, that's another national marine sanctuary, but further north off of, just north of San Francisco.
Did those last
Sonoma.
pieces go in?
I'm at full jam right now. I don't know if it's, I didn't see them go in, but I'll come off.
That's full.
Oh yeah, I think those little pieces might have been.
I think so. Turn it off and turn it on once. And still pieces coming, or is that it?
That's probably it, I think.
Okay, calling that good, I think.
Okay, that was sample number 84.
And Taylor Ann, do you need any water here to go with the?
If we could,
Um,
that would be great.
Yeah, let's go ahead and grab a Niskin bottle here before we move on.
Okay.
Oh, I gotta put it on.
I gotta switch camera a sec.
It fired, I think. I see
Fired?
that, yeah.
Thank you. Okay.
This is our first Niskin,
Yep.
first water sample. It all sort of blends into one. I think, didn't we already do one? But that was last night. Or the
Was
day
that
before.
085?
Yes. Sorry, I was writing down.
No, no, no worries.
Just gonna strafe those real quick before we lift off.
Yep.
6, 4, 7, 6.
Still going 045. Our next move.
Yeah, roger that. That'll be 045. Go ahead and call it in now. Bridge nav. Oh. 200 meters, 045, speed 0.5. Thank you.
See if we can't find some more rocks. It was a productive little outcrop of one rock.
Yeah.
Yeah, we got a lot done
going
there.
What is that?
I don't know.
Is that a nudibranch?
Getting followed by my dust. Still.
The sea cucumber?
Holothurian? Yeah. Never seen this one before.
I think it looks like Parasticopus.
Zoom if you want, Steve.
But I could be wrong.
Spiky.
Oh.
Very spiky.
Yep, that's a sea cucumber, Parasticopus.
Interesting.
So I say flush or two.
That's 2.
It's 2.
Flush.
Don't. Sorry, I know that's distracting. Trying to get it where it's in the middle. Super fast because we're shallow.
Yeah.
Yeah, quite stop it before it goes on the next Geneva.
Yeah.
So how do you get this sub-bottom profiler to mean anything, or is that all that soft?
That's
Does that mean something to you?
I know nothing about what frequency that bottom one is, if it's a separate one from the bottom tracking, which is the top.
But it's always been.
I've never seen anything useful come out of
Roger.
that. But I don't know if it's ever, like, we never needed it really. What's it say on the
I've played
0
with
to
it, but I
50?
can't get it to do anything other than
Yeah,
what you see.
I'll have to look what frequency it is.
I haven't read the manual lately.
Yeah.
Keep seeing some things that are floating across the seafloor, and I think they're big salps.
School of squid, or they other.
Oh, squid.
Coming up
I
by.
wondered where our squid buddies went.
Squid party.
God, they're fast.
And tasty.
Yeah.
Do we have porch light on or something?
Yeah, sorry. Port side's off.
I tilted too far up right now. It
Yep.
seems a little dark.
That will change with your altitude, but
Yeah.
It's that visibility up at the steps too.
yeah,
It's not as
and
good.
there's
Yeah, not as clear.
No crab there.
you can get super low if you want here.
Lily's pointed out that squid are one of the top 3 most valuable fisheries in and around the Channel Islands National Marine Sanctuary.
Yeah.
So she's not surprised we're seeing so many.
Troll scar.
Something big.
Yeah, so we shouldn't be seeing a trawl scar because we're within a marine protected area, a marine reserve.
Looks pretty old.
Oh, that was pretty old.
Okay. Yeah. Well, the marine reserves were put in place, I think, back in 2000, I want to say 6. So I don't know how long that would last.
Yeah, I guess it would depend on how deep it was and sedimentation rates and all that, but didn't look recent, whatever caused it.
That's a comb fish right there.
Don't combfish have, there's something pretty about them. Their dorsal fins, I'm trying to remember.
The long spine combfish has this long pair of like spines on the front of its dorsal fin that can lift up above its head.
Yeah, that's right.
And they usually have a pretty striking, pretty pattern on their sides too.
Yeah. Did you see it with the comb up?
No, comb down.
But yeah, they do have a pretty pattern on the side, the dots.
There's another one just to the left of the lasers.
You have a good eye, Meredith. Or a much better monitor than me.
Okay, I've got a really, it's not quite a 4K laptop monitor, but it's a very high quality monitor on the laptop I'm watching on.
Yeah, I'm going to switch my monitor's views right now. See if I can get a better.
I tried to watch one of the dives while I was at the office the other morning, and it turns out that my laptop monitors are much nicer than the monitor that's attached to my desktop at the lab. So I ended up running around the lab with my laptop so that I could continue to do lab work and also be on the science chat at the same time.
Oh, yeah, we were saying you were multitasking.
Yeah. And never sleeping.
I know. It's even when we're not on watch, it's hard to not watch and to not participate through the science chat because it's so so interesting.
So engaging.
Yeah, engaging. That's the word I was looking for. Yes. I've been known to take to turn everything off and then watch it from my cell phone in bed. So I don't want to miss
You
anything.
I
guys
have my
need
cell
some
phone
serious help.
Yes, I know. We're gonna have to go through withdrawal at the end of this.
I can
Go to
recommend a
some
good therapist.
Yeah, I was gonna say we're gonna have to go to some support groups.
You want to take a look at that shell stuff or go past it? Not interested.
Not interested.
We can take a look at it. Why not? Let's take a look.
Might have to hold you.
A lot more sediment up in the water column right now. I don't know if it's just blowing off in the current.
Zoom there, Steve.
Snails.
Ah.
Oh, it's a
Crab.
hermit crab fest.
Are they all
Are they
trading
switching
shells?
shells?
What? They might be in the line where they're all
That's
lined up
true.
trading shells. I watched a video
Yeah.
on that.
Oh, wow.
Lily Simonson sent me that video, one of the artists that.
House swap.
Yeah,
Yeah,
they
they're at
all
the
line up
marketplace.
and they put the shells in order, and then they all swap musical chairs.
Is there a reason for doing it?
Because they outgrow the ones that they're in.
Oh, okay.
So they line up in,
And
in size order.
Are you, is this true,
Yeah,
or
no,
are they fighting with each other over it? Do they really line up,
yeah, they do.
or they compare?
There's a clip from one of the, blue planets or
A
something.
time
like
lapse?
that.
Can we get a little more zoom on that?
Go ahead.
Yeah,
I'm not
Yeah, let me-
surprised that they would all congregate and sort of size up each other one by one, but that they would line up, that's incredible. That they'd be so orderly.
They appear to be lining up.
You mean like a conga line?
Yeah,
exactly.
that's like,
Yeah.
Yeah, they're fighting over
Two
that
of
one
them,
big
yeah.
shell.
Da, 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 da,
Yeah.
da.
There's two that want that bigger shell.
right.
Yeah.
I can't remember
And there's
the,
one already down in it. You can just see the little claws poking out.
He's like, no, this is mine.
I don't remember which, I'll find it.
Was it Life Story or something?
I recently sent it to somebody. I think I'll be able to find it.
When I was doing a scholarship program at Duke University, we did a study on hermit crabs in Panama and trying to see if they would follow certain sensory cues. So we put different cups of either crust conspecific shells or meat or different items of food that they would be interested in, and then counted the different types of hermit crabs that would come to each chemical cue source. And we noticed that the hermit crabs that had more intact shells would come around the crushed conspecifics to see if they could find a better shell. Or the ones that had ruined shells, rather, would come to the crushed conspecifics. And then the bigger hermit crabs that had shells that fit them properly seemed to be going more towards food or like feeding.
Oh.
It was interesting.
Yes, it is the Amazing Crabs Shell Exchange, life story on BBC Earth. It's a short
I'm gonna have
clip
to
on
check that
how
out.
they line up. So you said that, right, Steve? Life
Yeah,
story?
Life Story,
Yeah, BBC Earth life story.
which was on Netflix at some point. I don't know if
I'm gonna
it's still
try a
there.
button here. See if the button works.
You know, because we joke around so much,
I feel like it's more
I
jumpy
wasn't sure.
than it
Yeah.
was.
Less, I think you get a quicker command with the button, maybe?
Yeah.
Somebody's gonna end up without a shell.
I haven't seen any actual swaps yet.
Yeah,
But
lining up. They don't
They all
understand
look
the
very
concept of a
like,
line.
similar, like in size, except for maybe a few.
Yeah.
Like that one huge shell in the center. Everybody
Is
wants
that
it.
something they're feeding on in the center, or they're just
There's already a crab in that shell.
in
Yeah.
the big one?
Oh, that one's trying to get that one out. Clawing at him.
I just bookmarked it. I'll watch it after my watch.
Yeah, it's fun.
Oh, body slam.
Wrestling match.
Body slam.
Oh yeah. Gonna shake them out.
And the one that has already claimed the biggest shell is just like tucked in, like, nope, not coming out, not coming out.
Oh.
Yeah, that one
There's
was
a
the
lot
big
of
one.
energy being expended here.
Yeah.
But they probably have evolved some type of strategy where they only really sort of go for it with with an individual that has a shell size close to its own.
Yeah.
I mean, because what's the point?
It looks like the one in the big shell is too small for that. So they're like, get out of there, man. Let go of your ego. You don't deserve this shell right now.
Yeah, it looks like there's one right next to it at the top of the bigger shell that actually looks like it needs that
needs
large
it,
shell.
yeah.
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 Has all its legs sticking out there.
Chaos.
Funny, it's like they gang up to roll one of them over.
Yeah. They go in like a chain to pull it over.
Oh, he's coming out a little.
Get out of there. Small for that shell. Can't even move around.
Yeah, maybe that's why he's not moving yet. Because if you have the shell that you like, you just sort of take off, right?
I would get out of there if I knew I was
Yeah.
happy.
Huh, look at them grabbing each other and pulling each other off the sea.
That one pointy shell one's like, guys?
So aren't there sort of bedding rings for this? Like how people,
Who comes out with the top shell?
Yeah.
Yeah.
yes. I wouldn't be surprised if there was something like this on some island.
Gotta,
Not more with land hermit crabs, not with the
yeah.
sea ones, but
It's like, I wonder if the big one leaves, if there'll be a chain reaction. Oh, switch.
Yeah. That's what happens in that.
So have you counted
video.
yet? How many there are? I keep losing count.
I counted 38 or 40, but I keep losing count.
I counted,
just
like
got-
I counted 43.
I got 45.
40,
Wow.
okay.
I just got 38. I wasn't trying very
I guess I could cheat and pause the video, but
That would be cheating.
We can get the telestrator
Oh.
going.
Look at that one. It's got a good grip on that shell.
Whoa. Like a tower. All stacked up.
Oh.
That's not sustainable.
I don't see one extra empty shell, so everyone's going to have to trade into a shell and hope they don't end up with a shell that's smaller than the one they left.
Yeah.
so in Fiji, so it wasn't betting on who wins in this shell exchange, but they do have hermit crab races. I wasn't that far off. I mean, just watching this, I thought this is prime for human amusement.
Mhm.
Lizzie has said, well, although I could watch this all day, perhaps we should move on. And
Yeah.
we
Yeah,
probably
sorry,
should. They might
Lizzie.
just be waiting for us to move.
Voice of reason.
No, we want to stay here till the end of our watch. Make us move.
Put a little GoPro, see what happens.
Bridge nav.
That would be a great time lapse
Yeah, speed up that clip.
with a
Yeah.
Benny Hill clip in the background.
Yes. And I hope that this makes the highlight reel.
200 meters, 045.
The high frequency is 200 kilohertz. Low frequency is 20 kilohertz.
Yeah, I
So
think
the 21 should be the one on the bottom then, right?
Correct.
yeah, it penetrates the seafloor more, right? 'Cause
That's the
there's
idea,
less
yeah.
attenuation or something.
So our shipboard sub-bottom has a 12 kilohertz bottom tracker and then a 3.5 kilohertz sub-bottom.
And the gain is typically set around 20%, but is varied according to the water and target conditions and user preference.
Well, what'll be curious though is when we get to the harder stuff to see how that bottom one changes. What if you change the range of the bottom one to be?
So contrast and threshold sets the size and position of the receiver sampling window. It can help find small features in a generally featureless situation and exclude clutter from a heavily featured seabed.
The 200 kilohertz is crisp. Dialed that in. See if we can hand off the watches to some hard bottom.
So the first dB is the threshold, and the second one is the contrast. But if you hover over the buttons, they call it sensitivity and dynamic range.
Still moving 045?
045. Aye.
A pile of hermit crabs and then just back out into the mud.
Yeah.
Yeah.
They just sent out the hermit crab call. They probably used a shell.
Get some kind of little eyedropper full of whatever chemical signal that was, so you can just set up your own little hermit crab arena.
Yeah.
Yeah. And bring some empty shells along and just drop them in.
Yeah, it was really fun doing that experiment in Panama when we put their crushed shells in the water, just watching them all come. See what was going
Sorry,
on.
Rennie, I lied. The top one is the high frequency data.
Yeah, yeah, that makes sense.
There's a flatfish. Can't tell what it is from so far. Another sea pen.
Yep.
Is it the one that we've been seeing, the Stylatula?
Yep, that's the same one.
Stylatula. Another one coming up. Or several.
Yeah. You get fields where you get a bunch of them. I'm betting that there's more that are tucked into the sand that we're not even seeing right now. Working with the environmental DNA, it's always fun to see you'll get a mud bottom like this with nothing in sight, and you take a sample that's the supposed blank, and it's like, oh, well, the mud was full of sea pens, so it was sort of a blank.
Right. But so see, you were explaining also on one of the watches that some of these species shed a lot, so it might seem like there's lots of them, but because they shed so much. So these sea pens don't shed a lot of cells.
They don't shed as much as the big bamboo corals do.
Yeah, that
they'll
was
shed
the
enough.
one.
Yeah, the bamboos you can detect them actually from quite a ways off. I've looked at some samples from this.
Pick up the speed.
Sure.
Oh, I gotta pick up speed.
Yeah.
Looked
I
at
can
some
always slow the ship.
samples from the Central Pacific where there were a lot of bamboo corals around, and they got 50 meters off the bottom and took a water sample, and we were still detecting bamboo coral that high up.
Oh my.